Hello, welcome to the concise introduction into the science behind the stem cell treatment for lung disease using small mobile stem cells, as will be practiced in the future by SMSbiotech. I'm Abdulkader Ramo, the Chief Scientific Officer of SMSbiotech. I would like to take you through the forthcoming slides. The regenerative therapy is based on a single agent, SMS cell, that invoke several mechanisms for the aim of lung regeneration. The specific aim is to stimulate endogenous lung regeneration in individuals with destroyed alveoli, such as in the case of COPD. Chronic obstructive pulmonary disease, or COPD, is a progressive heterogeneous disease. It includes chronic bronchitis and emphysema, which is caused by the destruction of alveoli. The disease is caused not only by smoking, but also by air pollution, dust fumes, chemicals, and other factors, in some cases genetic factors. The disease typically attacks after 40 years of age. It has a significant effect on airways, becoming thick, clogged, and inflamed with a lot of mucus present, and the air sacs losing their elasticity and getting destroyed by that. This disease can be treated to lower the symptoms, but it cannot be reversed or cured. It affects 15 million US citizens, causing the death of 140,000 per year. It's a huge economic burden, and it also affects 400 million people worldwide. The main effect of this disease is, of course, the chronic lack of oxygen. The air sacs, or alveoli, are central to this chronic lack of oxygen. They are quite abundant and about 480 million in the lung. They occupy a large portion of the surface of the lung. They have a very thin wall composed of critical cells, the alveolar type 1 and the alveolar type 2 cells. Type 1 cells occupy actually 95% of the surface of the alveoli and are responsible for gas exchange. Type 2 cells produce surfactants that maintain the structure of the alveolus and are able to proliferate and generate alveolar type 1. There are other cells that are extremely important too, such as the endothelial cells that provide for the capillaries and the blood supply, and also the extracellular matrix, an important component that makes up the wall of the alveolus and maintains its structure and integrity. SMSbiotech is tackling the problem of alveoli degeneration and destruction through a unique cell that has been discovered by SMSbiotech, that is the small mobile stem cell. The cell is quite distinctive in many aspects, yet it has escaped recognition and detection for a very long time. Its size is about 4 to 5 micrometers, and it is fast and transparent with unique physical characteristics that distinguish it from all other cells. We isolate these cells from peripheral regular blood of adult humans. This can be done from individuals of different ages. The cells are translucent, stable, and quite resilient. They can be readily cultured and scaled up as needed. We can provide a large number of these cells using our patented technology, and we are currently producing a lot of these cells. The SMS cells are highly responsive to inductive medium conditions. We can control the cell behavior. We have years of experience in doing so. We can provoke these cells to produce extracellular matrix. We can control their movement and many other aspects of these important cells. SMS cells have a very important feature that is quite relevant to tissue regeneration. That is their selective interaction with cells that are already known to be key to tissue repair and regeneration. SMS cells have very strong interactions with key cells such as mesenchymal stem cells that are known to regenerate tissues, endothelial cells that form blood vessels, fibroblast cells where the SMS cells tend to inhibit in the case of in vitro, and a strong interaction, in this case, very pertinent to the lung, with alveolar type 2 cells. This interaction results in stimulation of proliferation and important changes in gene expression. Now, small mobile stem cells can be cultured for many passages, meaning producing a lot of these cells, but they can also be differentiated, meaning changed into other forms of cells, such as nerve cells, bone cells, and fat cells. It takes time for that to happen, but that really does not matter much with regard to the therapy that we are working at, because we rely on the interaction of SMS cells with other cells and not on their differentiation into other cell types. So we also know that SMS cells are quite able to self-organize. They are able to build a three-dimensional complex structure. These cells have a readiness in vitro to form complex organized structures. And these are really a good hint to them being involved in regenerative reconstruction of tissue and organs. One important aspect of these cells is their ability to produce extracellular matrices, to secrete these proteins that are critical for structural and biochemical support of other cells that make up a tissue. We know that SMS cells produce proteins that are already independently known to be important for tissue regeneration, such as vitronectin and heat shock proteins. We know also that the extracellular matrix derived from these cells promotes the formation of small vessels by endothelial cells. That we can demonstrate in vitro we can also demonstrate the formation of large vessels visible to the naked eye through the extracellular matrix produced by SMS cells. Those large vessels can be seen here in a video. The formation of these blood vessels from human endothelial cells are made possible by SMS ECM. This is actually unique to our technology and a statement to the capabilities that SMS cells present. The vessels can be isolated and studied further. We know that the formation of this vessel is central to any tissue regeneration. Without the blood supply restored to a tissue, the tissue cannot grow. It is important to be aware that SMS cells, when compared to other adult stem cells, embryonic stem cells, and iPSC, which are genetically modified cells, appear quite clearly to be quite distinctive. SMS cells present safety and efficacy. Indeed, they are superior to embryonic stem cells and induced pluripotent stem cells because of safety and the absence of ethical concerns, as they are derived from adult blood of adult humans and not from fetal or embryonic origin. They maintain continuous potency despite a lot of proliferation, something that is not achieved with other adult stem cells. They also have resilience to adverse conditions, and the small size allows it to be administered using a very particular item, that is the nebulizer. This type of administration is really unique to SMS cells. SMS cells attach to mesenchymal stem cells. Now, we know that mesenchymal stem cells are very important in tissue regeneration. We know how they modulate the immune system, lower inflammation, and start the regeneration of these tissues. So having the SMS cells bind to mesenchymal stem cells, stimulate their growth, and even change their gene expression ought to be extremely important indeed. This way, we can achieve endogenous stimulation of mesenchymal stem cells by simply introducing the resilient SMS cells to the body. This experiment where we demonstrate how strong that binding is to mesenchymal stem cells. We were able to detach the mesenchymal stem cells from the plates by enzymatic means, and SMS cells remain bound to mesenchymal stem cells. The strong binding with SMS cells indicates that the binding between the two cells remains there for quite a bit of time, and it represents a likely important biologic aspect of these cells. SMS cells also bind strongly to type 2 cells, not to type 1 alveolar cells. They stimulate their growth in the absence of serum. And that's quite important because alveolar type 2 cells that are known to restore the lung tissue are usually deprived of blood flow in damaged tissue. So being able to stimulate those cells using the SMS cells in the absence of serum is a great initiator for damaged and deprived tissue regeneration. Now, the SMS cells also change the gene expression of alveolar type 2 cells, and the changes in alveolar type 2 cells gene expression is consistent with the complex requirements for healing of the organ. The changes in those gene expressions, once studied in detail using references and proper software, indicate that SMS cells cause a lowering of inflammation, reduction in cell death like apoptosis, pathogen protection, formation of blood vessels, which is called angiogenesis, and less vessel permeability, and lowering blood pressure, something which is often accompanying COPD, that is called pulmonary hypertension. Interestingly, we also discovered, studying the gene expression changes, another critical aspect that is directly related to tissue regeneration, which is the active suppression of tissue fibrosis. If the tissue gets a repair that is based on fibrotic cells proliferating, you do not end up with an ideal tissue. You may get the tissue permanently damaged. So suppressing the tissue fibrosis and enhancing parenchyma cells is really what brings the tissue back. For regeneration means really bringing that status back. That is indeed what we observe regarding the changes in gene expressions. We observed downregulation of the genes that are responsible for tissue fibrosis and the upregulation, the stimulation of genes that inhibit tissue fibrosis. Away from these subtle gene expression mechanisms, the small size of these SMS cells, in addition to their resilience, made it possible for us to generate a mist using a nebulizer. And that mist includes the SMS cells and can be inhaled. Indeed, we can run these cells through a nebulizer, creating that mist, and maintain the full functions of the cells. Something that is unique to these cells because of their unique size and unique resilience, something that cannot be achieved with any other cell. So we will administer the cells directly to the lung and maintain the locality of the effect, so we don't lose cells to other organs. We can really concentrate the effect on the lung organ. We tested on animals the safety of the SMS cells using intravenous systemic injections. We also tested using direct application through the trachea to the lung. We used a large number of cells and we observed no adverse symptoms, no lesions, no temperature changes, normal appearance, normal behavior, no adverse effects whatsoever. In fact, we also tested whether these cells produce any tumors inside the animal. This is a test that the FDA requires us to do. We injected the cells not just in one place, but in many different tissues. We injected them subcutaneously, intramuscularly, intratibial, and in the lung cavity. No tumors were observed whatsoever. SMS cells are adult stem cells. They are safe. But what is most remarkable here is the fact that when these cells are injected into seriously damaged animal lungs, lungs that have destroyed alveoli, which is a typical model for emphysema, that is the standard animal model, we observed a highly efficient and quick lung regeneration. In fact, in 1 week, we observed almost 100% regeneration in the alveoli. The animals were able to perform as well as their controls in terms of velocity and endurance during exhausting physical exercise tests. So the upcoming cell therapy for emphysema, COPD in patients will be based on the unique resilience of these cells, their small size, when we administer them using a nebulizer. We will stimulate alveolar type 2 proliferation and causing, therefore, the production and regeneration of type 1 alveolar cells, therefore restoring alveolar cells. We will modify the activity of many genes related to alveolar type 2 cells that are critical to various aspects of lung repair and regeneration. We will also stimulate mesenchymal stem cell proliferation, reduce inflammation, and support overall tissue regeneration. We will stimulate blood capillary formation and provide access to blood for the damaged tissue. These are the main aspects and the cornerstones of this unique cell therapy. This is an extraordinary opportunity to invest in cell therapy for lung treatment. SMS cell therapy prospects for lung regeneration are really exceptional and not to be compared to any other available therapy. I will summarize it now. SMS cells will target several types of cells key to tissue regeneration, as has been shown in the bench many times. SMS cells stimulate the proliferation of alveolar type 2 cells that are key to lung regeneration. This is a given in the current research and understanding about tissue regeneration. SMS cells activate mesenchymal stem cells that are endogenous promoters of tissue repair in the lung. SMS cells will inhibit fibroblast cell proliferation, the main cause of lung fibrosis. Unlike mesenchymal stem cells, SMS cells are able to proliferate extensively without loss in potency. SMS cells are sturdier, more robust, easier to handle and administer. Their robustness allows, for example, the shipment of refrigerator temperature conditions to all over the world. SMS cells can be administered using a nebulizer and introduced to the lung directly, as we have seen. SMS cells are unlikely to induce any significant immune reaction because of the absence of the molecular targets for immune rejection. SMS cells are adult stem cells and are the safer alternative to embryonic and genetically engineered cells. The bloodstream is one of these cells' natural habitat, and that is unlike to mesenchymal stem cells. SMS cells demonstrate in vivo, using animal model, an extraordinary safety and efficacy profile. So we have multiple manufacturing advantages associated with these cells, and because of that, we are going to end up providing an allogenic, off-the-shelf, highly scalable production. The process of production is patented and proprietary. It's a production that is economic, meaning we can provide this therapy at reasonable pricing, centrally manufactured and shipped all over the country and worldwide. The shipping is at refrigerator temperature of 4 degrees, another unique aspect of this technology. SMS cells provide availability, stability, binding, effect at molecular and organ level, and a good safety profile demonstrated through preclinical studies. And soon, we hope, through clinical studies. So these human small mobile stem cells are a next generation stem cell regenerative therapy. There is no genetic manipulation required. It's a direct route to the lung. It's a multi-targeted, orchestrated, smart, intelligent, natural process. Off-the-shelf cell therapy, targeting chronic, widespread indications and aiming for cure, not mere treatment. Thank you very much for your attention, and please go back to me for any questions.